Thank you. That was great. And so I'd like, actually, don't sit down yet. I'd like all of our speakers to come up to the front, and we'll have a panel discussion. And I really think that there are a lot of interesting ideas to explore in this panel. So, you know, I love the buffet dinner that Dr. Hardy showed in one of his last slides as a good illustration of, you know, now we have a lot of things we can nibble on from various plates, and our challenge is to figure out how to put it all together into a delicious dish. And, you know, there were a lot of threads that came out of the earlier panel that I think it came up again here. Dr. Birnbaum mentioned the important foundational elements of data sharing and data repositories. And then, of course, we've seen just in this last talk how some of that is being developed and built out in this area. Chris Jennings spoke about the importance of common metrics and a common language. And this, the two panels actually before and after lunch, really interesting because they're very different metrics and different languages, but now all of the speakers are here together to think about these things in a more common way, and it's a challenge. And I think that one of the things that I've seen in the discussions in this workshop so far is that anybody who wants to work in the field of data integration will, for sure, be working outside their discipline and their areas of comfort and expertise. And the importance of working, therefore, in teams was mentioned by a number of the folks this morning. It clearly is important here. And it does make me sort of think about our field, the field of risk assessment and decision making, and how sort of, we do want to open the windows a bit here and get the air in from other fields, get some other expertise into the room. And I also just wanted to think back to one of Lance Waller's slides this morning, where I think this was a map of a typhoid fever epidemic, where if you just looked at where people lived and mapped that, you wouldn't really see any pattern. If you thought to map where they worked, there's a pattern that's immediately obvious. And so, how do we make sure that that especially with that sort of, it's a little contrary to some of what we've been hearing about needing to define the questions up front, because if we define the question too much, we may look too narrowly. So how do we balance defining the question and being open to the fact that if we define it too narrowly, we might be missing something? So before, sorry, the panelists, I'm sure have a lot to say, but I just wanted to mention there's a PowerPoint up here from a recent paper that was published in Mutation Research Reviews with Lauren Zeiss, who was on this Emerging Sciences Committee until recently, and myself and a few other folks, proposing to a little shift in terminology from G by E, which was mentioned quite a bit this morning, to what we saw as a broader I by E, in which I would incorporate all intrinsic factors, including the genome, but also the many other intrinsic factors that affect health. And then E refers not just to the traditional environmental factors, but also to dietary, socioeconomic, and microbial stressors, which some people define the exposome that way, others define it more narrowly. The idea would be that this framework could help integrate two risk assessment challenges. One is around cumulative impacts, and the other is around susceptibility. But this would be massive data integration exercise, of course. And actually, along those lines, it seems like the Plato software that Marylyn spoke about might be kind of moving in that direction. So maybe I'll start with all of that intro by starting with Marylyn for your reactions and some more thoughts about where that aspect is going with Plato.
Yeah, so when we first started developing the software, So it stands for the Platform for the Analysis, Translation, and Organization of Large-Scale Data. You know, every software needs an acronym. If you just said, like, this is a tool to do stuff, nobody would use it, so it had to have a cool acronym, so Plato was it. We developed it initially for genome data. We really started it back in 2005, 2006, and the genome data sets were getting bigger, and we needed a way to analyze them more efficiently than SAS or S+ or R were able to do. The data sets were just too big for that. Well, then another genomics tool came out around the same time called Plink, and so we said, oh, well, we can use Plink for that, but in addition to all the genome data, we also have all this clinical data and all this environmental data and all these other categorical, binary, and continuous data on patient samples or individuals samples. And so we thought, well, we just want to take that framework and extend it. And so the analyses in there are your very standard linear regression, logistic regression. We have some interaction models where you can do the full model and a reduced model and a likelihood ratio test between the two. We included Firth regression. And really, it's just a matter of choosing, you know, do you have a binary outcome or a continuous outcome, kind of your dependent variable, and then which types of independent variables do you want and how do you want to test them? And so the software itself, I think, can scale for, I just wrote that down. I love the I by E, because when I talk about the genome, I really think of all the ohms and all the biologics that we have and could measure internally. And when I think of exposome, I think of all the exposures, the social determinants of health, the psychological determinants of health, which are partially I and partially due to E, as well as, the microbiome, that's kind of partially I, partially E. So I think of all of those. So that was part of the motivation for the software, was that, you know, in my group, we really think that we're studying complex traits. Kind of most of the human health conditions we're looking at are complex. And by taking the reductionist biologist approach all the time, like, that works for certain things, so I'm not totally knocking that strategy, that having a hypothesis and specifying a model and testing it works if we have enough other data to tell us what those are. But so much data is out there now that we have to do some fishing in order to give us some models to then go to other data to test. Actually, I forget if it was Chris or, what's your name? Bruce, this morning, that had are we doing a fishing expedition or hypothesis? I think it was Chris that had that, right? Because I think we need both. So if we don't do some fishing with all of this big data to narrow down some elements, we can't do the modeling in another data set and do the hypothesis testing, the hypothesis-driven hypothesis testing. There's just too many. And so, so that was the motivation for the software. I think it can do those things. We're still trying to add kind of some other computational, like other statistical models, not just regression, some of the non-parametric models, but kind of so much of what the journals want are the parametric models. Even if you do lots of really cool machine learning, the reviewers will come back and say, what did you fit that with a regression model, and what does it look like? And so we kind of started with that, even though we do a lot of other machine learning stuff as well.
Great. And so my next question is for the risk assessors. Maybe I'll start with Chris about all of this information generated in EWAS studies, G by E or I by E, as the case may be, type of research. Do you see, in looking toward the future, any of that information being usable for more than hypothesis generation or maybe sort of helping with hazard identification? Can that information maybe take us further in the risk assessment realm?
I'd like to think it could. I mean, I think right now it does seem like it's more utilized in the hazard, often sometimes in more the supporting evidence kind of role. But I think the more, especially the more it becomes informative in terms of at least features of the data for sort of dose response, then I think that sort of changes it. And I think that there might be ways that we have to think about evaluating the quality of the evidence, but I don't think I think it's addressable.
Right.
There's probably conversation to be had about what that looks like, but I'm sure something could be devised.
And, Chris, let me pull you in and let you ask a question, and then I'll move on with some other.
So I'm constantly interested in this idea of looking at all these different chemicals. I think the EWAS and the GWAS and all, it's like one at a time, but we're doing a lot of them, right? How do we incorporate, my bias is that I think in environmental health, we really need to get the whole exposure down, like not just one chemical at a time, but all of it down, right? So we need to start thinking of it integrated. We need to start thinking of them as mixtures. We need that we're all exposed to all of these things. If we're just looking at one at a time, so how do we kick it into another whole way of thinking, and not to leave behind the let's look at one thing many times, but we know a lot about multiple.
You are unmuted.
Oh, Atul's on. Great.
So anyway, I think you know what I'm trying to get to. How do we change the story? Is what I'm trying to ask.
And can I add to that question or build on it a little bit? So I think this is a question for Tim Pasteur and about the Risk 21 concept. And I think going back to this I by E concept, you're looking at hazard, you're looking at exposure, but you're also looking at all of that against a background. And that background includes not only other chemicals, but also many other susceptibility factors. And so how do we incorporate that kind of complexity? I didn't see a lot of it in Risk 21 so far, but maybe that's coming.
Yeah, Chris, your question is absolutely spot on. And frankly, I think, and those of us in the Risk 21 community think that this is probably one of the biggest challenges we have in the next foreseeing years, is how do you actually go about doing a safety or a risk assessment on multiple chemical exposure? We do them one by one. We get lots of cool data on individual chemicals, but the fact of the matter is that it's a combined exposure. Now, we've got 2 papers in the 8 that I showed you that are trying to tackle that problem, that issue. One's a problem formulation paper, and you can go through that and kind of get an idea of how you frame up whether you'd even need to address it or not. The actual framework that was proposed by, again, I can't remember, it was probably about 15 different authors, is looking at gateway steps in deciding what kind of information you need and then progressing through that. My view on it is that is a first step, along with what EPA and EFSA have done with trying to crack that nut. I mean, we're trying really hard to get at the whole nature of the human exposome and what that means in terms of safety and health. So it's a big question, and I'd say we're building that here, starting with that.
Great, sure. Marylyn?
I think the same thing is happening in the genome space, and so we've been thinking about this on both the exposome and the genome, that one, either we need to use multivariate type methods that are taking the multiple exposures at the same time into the model, and whether that's additive or interactive or both, that's important. Or doing some sort of data reduction, like is it that we have multiple chemicals and it's not the interaction of them, but it's actually some sort of like it's this or this, but actually if you have this, it's a good substitute for that. So can you do some sort of principal components or factor analysis to pull out the elements that are important to then model, or using machine learning technologies? And part of the challenge around all of that is that we tend to go back to our we need to do a p-value assessment at the end, and we have to Bonferroni correct for the number of things we looked at, and we ignore the fact that they're all correlated. And when you do these interesting multivariate interaction models, they're not Bonferroni significant usually. And so we have some models of interactions that we're like, oh, that looks really interesting. But there's one, we just finally got it accepted for autism risk. It's copy number variance with ozone exposure. But the p-value was marginal because interactions use more degrees of freedom, and it's harder to get a Bonferroni significant p-value, and it was really hard to publish. And the reviewers kept saying, well, but is there really evidence for these types of interactions? That was just one exposure with the gene. But I think you're right that it's not only the interactions, but it could be Boolean function. It's this and this, or this and this, or these 3 other things. And so machine learning technologies could get us there, but we're not at the point that we can just use them yet.
And so it seems that this is raising the issue of biological plausibility, which also came up in Chris's talk, and the sort of, what is the biology telling us, and some of the questioning is when you start sort of finding things where we don't know based on the biology, if it's pushing new frontiers. And so I know we have Atul Butte here with us in space. And so I'm actually interested in his thoughts about using the electronic health records and whether that clinical data integration component can help us at all with understanding the sort of the biology better as we then move into risk assessments and decision making.
Yeah, so I'm guessing you guys can hear me. In the end, I think that's exactly right. I mean, the electronic health record data, one view that you could take is that it's an incredible view of this human physiology, right? I have all these pulse rates, and I have the blood tests. But again, we're also limited by the blood tests that people actually order, that physicians and nurses actually order. So we might have lead levels on a bunch of kids, but rarely past that in terms of age. So I think it's great, but it could be limited. Of course, there are ways to get through samples from these patients, right? And biobanks and the precision medicine Oliver score is one example of a good one, a big one. But in the end, I think, to get to the deeper question of what does prove actually mean, right? What does it mean to actually prove something? And you can go after p-values and you can go after statistical significance. But I think in the end, two ways I think we're going to want to increasingly think about how to prove something. One is to look for orthogonal models, and I know it's hard to find an animal model or a mouse model for a lot of these environmental factors. Never mind 2 of them or 3 of them all happening at the same time. Modeling that is incredibly hard. But the other kind of way to prove something, I think, is going to be with an independent cohort. I get a p-value, and then here's another separate cohort, separate part of the world, separate health system, and look, we're finding the same thing. Or it's a different cohort across time. And we've done that in Hayes and a couple others. So I think this level of proving and biology, and yeah, it's great physiological data. As great as an electronic health record repository is, you kind of want 2 of them or 10 of them, right? All independent, so you can kind of learn from a few and test in a few others, I guess.
Yeah, that's very helpful. And and speaking of this issue of, it seems like there's a lot of different issues of scaling here. And there was a question actually from the web audience about how do we deal with the different sort of all the different scaling of all of these different databases, temporally, geographically, and different dimensions. And so I'd actually like Sandy Eckel to weigh in about how she sort of thinks about that, because you've really grappled with that issue.
So I do agree that it's really an open question at this point. I've been primarily thinking temporally because that's kind of what I'm most familiar with, dealing with time series studies of air pollution. But I think it's very important to keep in mind, what is the scale at which biological responses happen versus what's the scale at which we can measure something. Because I don't know that it really will be relevant to measure air pollution exposures, at every 10-second resolution. I think, maybe we should try to optimize our measurements to better correspond to the biological response, but then that assumes that we know the biological response. So it's a bit of a circular, kind of getting to Lance's earlier slide, it's kind of a circular logic of we can't design optimal studies until we really know what our question is. And so I think it's going to be an iterative process.
Okay, I see a number of hands up. So I'll start in the back and then come around this way. Yes.
My question goes to Sandy. Considering the reliability of data, you're saying that 1 in every 12 Americans is having a problem of asthma. So when you consider the number of diseases, cancers, tuberculosis, cholera, etc., is asthma the most important ailment in America? And also, I want to say that when you look at the facility required in terms of human facility, in terms of drug and a lot of things required to handle this issue of asthma, have the concern that asthma, were you able to isolate it from other diseases before you establish your facts that asthma is 1 in every 12 in this country?
So I do agree that, asthma is just one disease, but I think okay, so I'll try to repeat it as I understood it, and correct me if I didn't understand the full depth of the question. But so what I was understanding is that the question is, asthma is just one disease. There are many other important diseases out there. Why start with asthma? Why are we investigating this question in particular? And so I think that we're starting with asthma because it is an important health problem. There are many others, but there are some really relatively well understood triggers. And so if we can, have this paradigm of studying triggers using sensors and sort of start to validate our understanding that we have now, I think that we could apply that to other areas of medicine as well. And, the idea of patient engagement in managing their own disease, this is a model that could be used, in diabetes or other very important areas as well. So did that cover your question?
Great.
Important disease in America and also.
I think, I don't know that we need to talk about
Because
what disease is the most important one, but I think
was
the
pre-
point at this workshop
No.
is to look at the tools for integrating data.
Yes.
And so I think that, one of the key issues that came to me from Dr. Eckel's talk was really how they've been integrating data on physiologic measurements and environmental measurements over time. And that's super important. And related to the topic today. But I know that Lance Waller also had his hand up. So do you still have your
I still have my hand up. Okay.
thought so?
I have an electronic medical record question that I'm afraid to ask. So folks for Atul and Marylyn. And part of the reason is I don't like to ask this of my own IRB. But Atul showed the IRB protected health information for HIPAA, which said no geographic resolution below a 3-digit zip code, but showed 2 maps of zip codes of all his patients. And I believe, are those 5-digit zip codes? And then, the point about, I do a lot of spatial analysis, GIS. I don't want to ask my IRB, can I use census tracts, for instance? Can I use this? Because I hope that's de-identified enough for what I do, but to be honest, it doesn't fit the HIPAA rule. So I'm just curious in how what's your experience with IRBs and your own view of that?
Yeah, so it's great, and of course we have to follow the IRBs for research. And you're right, I didn't make this at all clear. First of all, we have a business reason to integrate this data, right? So we have this ATO. We're trying to understand our variation in care. We're trying to improve our healthcare quality. So those are all reasons that we can have an identifiable database in the middle, okay? So we can actually have that identifiable data. And from there, the idea is to make a de-identified version of this database. So you're absolutely right. To de-identify this data, we would be removing, so you can do zip codes, except for zip codes with small populations. There's a list of those in the rules. Census tracts a little bit smaller resolution. So you could make a limited data set with that. So you're absolutely right. For research purposes, you're absolutely right. We would have to de-identify that. But because we have a business reason, a quality reason, our central database is actually identifiable data. We do have the locations there.
So for mine, it's
Can I
research
just push
only.
back on that a little bit?
Oh, sure,
I mean,
go
just
ahead, and
having
then I'll give you the
having a
research one.
business reason. I don't know. I think there's more to it than that. But I'm sorry. I think I'd like to discuss maybe at another time. I'm not seeing quite the point, but anyway, it's something I struggle with. I mean, I want to have as fine a geographic resolution as I can because I think I'll get to Sandy Eckel's point. There's a scale at which stuff happens. I want to measure at that scale. I have my best chance of finding it. But once I incorporate the patient data, now I'm under HIPAA rules. If I'm only using the census data, I don't have the health outcomes, so I don't have to deal with it. But It's a challenge.
So there are two ways around it for doing research. One, well, I shouldn't say ways around it, ways to deal with it appropriately. So one is some institutions have set up a data broker where there is an individual kind of in the middle. So you have the identified HIPAA-protected data, and then you have the researcher who's behind a firewall. And you have a data broker in the middle who is allowed to see the HIPAA-identified data, and then they can do the de-identification to provide it to the researcher. So the researcher never sees the zip codes. What the data broker would do is take the addresses and zip codes, do the GIS mapping, do the linkage with whatever database the researcher wants, and give the researcher, here are your patient IDs, and here are the variables from your databases for those patients based on their geocodes. So the patient or the researcher would never see the actual HIPAA identifier. They have kind of a middleman that does the processing. And the data broker doesn't have access to the genomic data or the whatever other data they're going to be doing the research on. The data broker doesn't have that and isn't looking at that. The other way is to apply for an IRB application that is identified and with a HIPAA waiver, where you would say, this is the HIPAA identifier that I want to use, here's why. When you do so, you have restrictions on what computer system it can be on, so it has to be in a HIPAA-compliant computing system. I've actually been just talking about doing this at Penn, and they said as long as, so I could have somebody in the research group be that data broker and do that part in this kind of safe space behind the HIPAA-protected firewall and do that mapping, but then when we take it into the research lab to link with the genome data, it'll have to be already mapped and coded. So there is a way to do it appropriately. It takes extra work, but I, like you, I think it's worth those extra hoops to go through because I think that information is going to be incredibly valuable. It's going to be a lot of work to get it mapped to the point that we can use it. Part of the challenge, too, to your earlier point about we have to get in these areas we don't know. Like, I don't know spatial statistics. I don't know GIS. Like, I'm actively looking for collaborators who can help me make sure we do that right, because to do it wrong is going to be useless.
Yeah, just to close out, there was a discussion in the first panel, too, that I think the broker, the data honest broker works well until you get to really small numbers in small areas, and then you've got the count is one. So some agencies and data sources will resolve, they won't release anything below a certain number, which leaves some holes that you have to model around too. So I bring it up that there's some aspects of privacy and confidentiality following the earlier discussion. I don't necessarily want that to be the only thing we talk about, but thanks for the details from both of you on the aspects of this, just because it's something we run into a lot. So thanks.
That's important. Wei Shui, you had a question or comment?
Yeah, so I wanted to return back to it was inspired partially by this morning's panel about the idea of needing to confirm, and not just we just talked about the hypothesis testing versus hypothesis generation, but also related to the sort of integrating across different data streams that Chris was talking about, because there's been a lot of talk about how do we get all the human data together and all the different aspects of human data, but how about all the other experimental systems? There are also biological systems, and my impression is right now a lot of the toxicology world is focused on it's sort of a tox-centric ontologies, right? And now we have these human-centric ontologies or structures, but how do we put those together? I guess I see that as a big challenge that hasn't really been touched on, other than kind of a more, we look at human data, we look at these three data streams, and when we try to integrate it on an expert judgment basis, but is there a sort of more data-centric way to integrate these sort of different types of data, essentially, going beyond human to the other in vitro and experimental in vivo systems?
Okay, I'll let
the only
Tim
thing
start,
I'd say
but
to that
Dr.
is,
Hardy may
it's such
also
an
have
interesting thing to watch a toxicologist versus, say, a physician look at it, because a toxicologist says, what diseases would this chemical cause? And then there's this array. A physician might look at it more from the standpoint, I have a disease, asthma, and I want to know what causes that. And what you're saying, I think, Wei Shui, is that how do you bring those two groups together and make sense of it in a fashion that we can then see that together? You expect me to give an answer to that, right? Great question, but I think that's what we're here trying to build.
Right, so I guess maybe having a common data sort of ontology or some sort of language that bridges that, because I think it was talked about this morning a lot and also you talked about the language barriers between these two communities, I think are a barrier to integrating these data in a more structured manner rather than currently it's kind of each siloed has their opinion, and then they kind of get equal weight at the end. We put it together and say, well, it ends up here because the toxicologists say this and the epidemiologists say that. And so, but is there a better way to integrate that?
I guess we're thinking about that in Hawk. I mentioned for, I think, the animal study, there were 4 levels at which you could describe an endpoint. Actually, there's 5. And we have the same for the epi fields, which I think is sort of really helps bridge the disciplines. We're definitely trying to sort of reach out and sort of find the terminologies. But I think we might have to accept, because, a lot of these systems are already developed. We're probably not at a point where we can sort of say, okay, we're all going to call this. But I think sort of synonym-based approaches, so maybe you sort of give a little bit of flexibility across the databases, but you can find things. So, but I think especially on the, at least within IRIS, on the epi side of how we capture information, that's something where we're keenly interested in trying to bridge with more of the clinical community.
Just one last comment on it. We published a paper some time back about bringing tox and epidemiology together, and that was published in ToxSci. Gary, I think, was part of getting that published, because we sat down with epidemiologists on one side of the table and toxicologists on the other. We had food fights over the whole thing, but it's all about epidemiologists bringing levels of evidence or causality to the table, and toxicologists bringing quality data to the table, systematically reviewed, that you can then pair up in some visual framework. And we had that as well. So that's really what we're trying to achieve. And I think each one of these little pieces of the puzzle are going to fall in place as we keep asking those questions, publishing the papers, and doing the work. Linda?
Linda.
Okay. So I think one thing we have to remember is that when we're looking at clinical data, we're looking at individual people. When we're looking at epi data, we're generally looking at population data. When we're looking at tox data, we're looking at individual data, basically. And we have to work on ways to bridge, because what you may see in an individual, you may never, or you may see things in a population with shifts of distribution that you are going to be impossible to see in an individual, except, and someone was talking about this importance, the importance of looking at the extremes. I mean, that's why the extremes are so valuable, because you can see things that you never see when you're just looking at the mean.
And, okay, sure.
This morning, I almost asked the question during the panel to Lance and Chris, because that is the problem. Like the fallacy of so much of what we do right now is that we believe and we are taught that bigger sample size is better. So big N. Power equals sample size, which is not true. Power is sample size and effect size. And so by adding variation and variability, you're decreasing the effect size of each of the effects and therefore decreasing your power. But the belief is big sample size. Some people know better, but kind of the general thing that people remember from Biostat 101 in high school is bigger sample size is better. And then the other is that yeah, I had it in high school, end of high school, senior year. And then the other is that kind of the main statistics that people remember are t-tests and ANOVA, which are testing group mean differences. And so we collect these big samples of people, and then we look for correlations and associations. And some of them are related to the mean. Most of what we're really interested in are the really rare things. All of precision medicine is about the N of 1 or 2 or a few. It's really rare. And then we get super upset when we do this big study and we look to see if this has an effect, and there's no effect. Well, we did a study of the population and looked to see if it affects the mean, and it doesn't affect the mean. It affects the 35 people in this one tail. and your t-test or your ANOVA doesn't find it. So somehow we need to wrap our heads around kind of changing our paradigm on sample size and power, that having the right sets of individuals or data is better than just all of it, and not just testing the mean. We have to test the variance or test the tails, something different from just t-test of the mean.
Just one follow-up, I totally agree, except that at times, knowing about the mean is very informative. You know, so again, as someone said it this morning again, what's your question?
Melissa and then Kim.
Melissa Perry, George Washington University. One other important distinction, in addition to individual versus population, when we're talking about epidemiology and toxicology and precision medicine, we're talking about observational data versus experimental. So we're talking about dose versus exposure. So there's a deliberateness in a tox study with animals. So we have to be very aware of that as well.
Okay, then, sorry, I wasn't.
Related, I think some of the big successes we see with data science, though, are the things that we all think of, like your Netflix, your Amazon, whatever, which are extremely tailored to the individual, correct? Right? They're not trying to make a statement about what a human being would choose. They're asking what you would do, right? And so some of these algorithms that we're applying them to big data. I don't think that we should be surprised that the outcomes we get are sometimes not what we expect, or we all have funny Gmail things that it's thrown at us, or Amazon suggestions based on something, right? But it's the individual, and we tolerate so much error with those things, and then we port those same methods to something where it's not okay to make a mistake like that, or a population, and are sometimes surprised. So I think there's also actually a lot of methods work to be done on the actual, maybe boring, maybe exciting, depending who you are, actual methods stats there as well, beyond just these application ideas.
Okay,
Follow-up
now
comment?
going to Kim.
Oh,
Oh, no, not Kim.
So
I'm
I just
sorry,
wanted to make
Sandy.
one quick follow-up comment. I really like that point about individualized models, and how I think another comment along that line is, imagine that we could build beautiful individualized prediction models. How would we draw information from that? How would we because we do public health at a population level. How would we get information from all these individual level models?
All right, Kim.
So Kim Bucklehead, Brown University. I'm sort of going to ask a question that steps back from the individual pieces of what we've been talking about and compares the case studies, the two case studies, clinical outcomes versus risk assessment. And I get the impression from hearing those case studies, first of all, clinical outcomes people have much better language. They're doing precision work. That there's a lot of certainty in the clinical outcomes world. You're dealing with huge numbers, 14 million people. You're dealing with humans. You're dealing with diseases, which are, things that are real, physiologic, obvious kinds of endpoints. Genomes, where you can quantify lots of stuff. So you've got highly quantifiable data with lots of people, and therefore what appears to be a lot of certainty. And with risk assessment, we're mostly using surrogates for humans. Our exposure levels are very low. The effects are unobvious until you go to high concentrations in some of the models. And there are lots and lots and lots of different data types that are surrogates. So there's a high degree of uncertainty. So I just wonder what the panel, if you reflect on that, what you think of that difference of what we've been talking about.
Tim?
Well, in my view, toxicology is doing nothing other than establishing a degree of plausibility. Of course, it's a dose response as well, but it's establishing a degree of plausibility for what you might see in human populations. And I think Wei Shui or someone mentioned that it's the difference is whether it's a dose or whether it's an exposure to humans, dose to the animal in a controlled fashion. So really what you're doing in the situation of toxicology and trying to pair that up with epidemiology is providing a sense of what would be plausible in a biological system. As a toxicologist, I always thought for a long time that we were the key to success in risk assessment, but I think it's really down to a plausible biological effect in human populations that could possibly occur at a given dose. And that's about the best we can do at this point, given the fact that they're surrogates for human beings.
I think there's only time for one last question before the break, and I may take the moderator's privilege to ask that question because Tox 21 and ToxCast came up a couple times in the morning discussion. I think Chris mentioned, well, in the context of some of the EDC work, and Linda also mentioned it. And there's a lot of data that are being developed that are actually pretty big data, querying toxicity pathways in human cell lines to identify chemicals that may be biologically active in those pathways. So to what extent could those results then be integrated in potentially to electronic health record data, as just like the genomic data have been, or to see if the chemicals are associated with perturbation of the same precursor pathways in humans? Is there some kind of line of inquiry there that panelists think might be worth pursuing? It doesn't. Nobody seems to be working in that area, and so if there is someone, then that would also be good to know. Or is that too far out of left field? Oh, wait.
At A&M, we, my colleague Yvonne Rusin, he's been
he's
doing a
some
lot
work.
of work on iPCS-based derived differentiated cells, cardiomyocytes, heart cells, neuronal cells, liver hepatocytes, et cetera. And so, we're doing TempoSeq to try to get at the gene expression changes. And so those, in theory, there's a common language with the human clinical genomic epidemiologic data as well that could be tied in. And because it's in vitro, you could potentially test more than single chemicals as well. You could do chemical mixtures. You could take actual environmental samples, take extractions, and then test those. So, there is potential maybe for some in vitro quasi-validation of things that are observed through this sort of big data, through those type of approaches.
Yeah, because I think originally in the Tox 21 committee, there was this thought that some of those omics assays or pathway-based assays in chemicals would help to identify chemicals that might be associated with certain types of endpoints, and then those might be then evaluated in like worker populations where there are people exposed to them. And so I'll let you have the last word, Dr. Hardy.
So when you go to look at this kind of biological data, you find there's a lot of variability. You change the dose, the omic signature is different. How do you interpret that? And so it needs to be embedded in our social way of doing the research. It's iterative. So you do some kind of pathway analysis. It needs a knowledge-based analysis to make sense, but it has a lot of uncertainty. But that needs to feed back into other people on your program. Could you run those functional assays and verify if this assumption I'm making on the signature is okay? Is it consistent with what the ToxCast assays are saying on endocrine disruption mechanism? And so I think there's a lot of importance of feedback loops that are knowledge-based, which means I don't think it's just a data integration problem. We need to put the data on the table, but there's always a lot of need for discussion, and we need a common ontology for a common language, but it's a common understanding and a verification and a feedback loop we had, which was part of the theme I wanted to add to the recommendations here, that education and cultural interaction are really important, in addition to all the wonderful things people are doing with data science.
On that note, thank you all very much for a very stimulating set of discussions.